Let's now look at the regulation of glycolysis. There are 3 steps in glycolysis that are regulated. The first one is upon entry of glucose by the enzyme hexokinase to form glucose 6-phosphate. This, of course, is the trapping reaction and also preparing the glucose for subsequent reactions in glycolysis. And remember, this is part of the preparative phase, so we are using ATP as a source of phosphate for this reaction. And this reaction is very simple to regulate. The hexokinase is undergoing a feedback inhibition from the product. So as the glucose 6-phosphate starts to build, it will feed back onto the hexokinase enzyme and slow the process of glucose conversion. Now, when we're talking about skeletal muscle, or red blood cells, like we did in the glycolysis video, considering regulation at this level is very much unimportant. And the reason for that is that the skeletal muscle is taking up glucose only in the fed state, responsive to insulin signaling, which is translocating GLUT4 from intracellular vesicles to the cell surface. So the GLUT4 is taking up glucose for the primary function in skeletal muscle to generate ATP and for storage as glycogen. So there's no real need to be regulating hexokinase at this level. This will become very important in the liver. Red blood cells, of course, have GLUT1 on their surface. This is not responsive to insulin. It's on the cell surface at all times. It is a high affinity transporter for glucose. And so glucose is always available to red blood cells under any conditions of plasma glucose levels, whether it be fed or fasting state. For red blood cells, this is primarily going to be translated into ATP synthesis through glycolysis. Because red blood cells do not have mitochondria, they are going to take the final product of pyruvate and convert it to lactate. So red blood cells are essentially, under these conditions, anaerobic. And this makes sense because they're oxygen carriers. Their job is to deliver oxygen to the rest of the body, all other tissues, but not use it themselves. The next level of regulation in glycolysis is the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. This, of course, is another preparatory reaction in glycolysis, so we are going to be using ATP at this level, converting it to ADP, and the enzyme that does this is PFK1 for phosphofructokinase 1. Now this enzyme is considered a gatekeeper. It is a gatekeeper in glycolysis because it is going to regulate the flow of substrates, oxidation of glucose, going through glycolysis. Or if we inhibit this reaction, we can form glycogen. So recall this particular reaction from glucose 6-phosphate to fructose 6-phosphate. This is done by the enzyme phosphoglucoisomerase. Now, if we recall our thermodynamics, the delta G of this reaction is approximately 0. So this reaction is readily reversible. This reaction, previous reaction, is not reversible. That is an irreversible reaction. So if we regulate PFK1, we can increase the concentration of fructose 6-phosphate, which will tip this reaction into a negative delta G going backwards, being exothermic and favorable. And now we build up glucose 6-phosphate because this is not reversible with the hexokinase enzyme, and we can use this for glycogen storage in skeletal muscle. So what regulates PFK1? PFK1 is essentially an energy sensor in the cell. So as ATP levels begin to rise, and especially in skeletal muscle, the ATP will bind to PFK1 and inhibit. So once the energy charge inside the skeletal muscle is as high as it can go with high levels of ATP, the skeletal muscle will say to itself, let's store glucose. So if we inhibit PFK1, we increase fructose 6-phosphate, our product to substrate relationship changes, and the delta G becomes negative free energy going in the reverse direction for glycogen storage. Now likewise, as ATP levels start to drop, AMP levels will begin to rise. So as we are using energy in skeletal muscle, we want to start PFK1 again to proceed through oxidation of glucose and generate energy in the form of ATP. So AMP is a positive regulator, an activator of PFK1. Now let's take a look at why this is the case with these levels of ATP. If we look in the resting state, R being rest, A being active, the resting state of our skeletal muscle ATP levels is at 100%. This is ATP. Now if we become active, the concentration of our ATP in our skeletal muscle will drop to, say, for example, 80%. Now this is for ATP. Let's look at ADP. At rest, there's typically a small amount of ADP in the cell, just from normal activity. But under active conditions, we convert ATP, dropping its concentration, to ADP. So we have a small, measurable change between rest and active states in ADP. Now let's look at AMP. AMP at rest is 0. In the active state, AMP becomes measurable. So although ATP, there are small fluctuations, ADP, there's an increase over the basal level, another small increase. This is essentially an on or off switch, meaning it's not present in a resting state and it is present in an active state. Hence why AMP becomes a very important, powerful regulator of PFK1. It's not sensing small differences, it's sensing whether it's there or not. Now how does AMP come about? Well, when we have activity in the skeletal muscle, we have the conversion of ATP to ADP plus organic phosphate, and this is, say, due to contraction of the skeletal muscle. As this reaction proceeds and the concentration of ADP increases, there's an enzyme called adenylyl kinase that becomes active with increasing concentrations of ADP. Adenylyl kinase will take 2 ADPs and convert them into 1 ATP plus 1 AMP. This AMP is what appears during an active state when we're using ATP, and it's also the sensor that is activating PFK1 to allow glycolysis to proceed so we can get the ATP levels back up. The final step of regulation is the last step of glycolysis. We're going to convert phosphoenolpyruvate into pyruvate, and this is done by the enzyme pyruvate kinase. This is importantly one of our ATP generating states reactions, so we're going to convert ADP to ATP, harnessing energy. And this enzyme, pyruvate kinase, is positively regulated in skeletal muscle by fructose 1,6-bisphosphate. So fructose 1,6-bisphosphate is an activator of pyruvate kinase. This is essentially a feed-forward reaction. The importance of this is because all of these reactions in glycolysis between fructose 1,6-bisphosphate and phosphoenolpyruvate, the delta G's, the free energy changes, are all near zero. So that makes all of these reactions reversible. So if there's any slowing in glycolysis for some reason here, the reverse reactions could potentially occur, which will throw ATP production off. So in order to prevent all of these reactions from being reversed, we are going to take the last step in glycolysis and have a feed-forward reaction to increase the activity of pyruvate kinase. By doing that, we essentially deplete phosphoenolpyruvate. Again, this is abbreviated PEP. We deplete this substrate, converting to this product, so that we continue a negative delta G change from fructose 1,6-bisphosphate on down to pyruvate.